yeah, thanks for the invitation. Thanks to all of you for coming. It's nice to have an in-person audience. I'm really tired of talking to a computer screen, so this is a pleasure. So today I'm going to be talking about work that I've done to use genetic tools to help conserve and manage rare midwestern plants. Okay, so just a brief outline. Let's see. No, it's not doing it. There we go. Okay, so as an outline, first I'll just give an overview of conservation genetics and the program that I manage at the Missouri Botanical Garden. Second, I'll give you some examples of how genetic data can be used to make conservation efforts more effective. So the first project we'll talk about using genetic data to devise conservation strategies to protect the genetic variation in rare species. And then the second is genetic study to understand the relationship between genetic diversity and reproductive success. Okay, so just to start off with, though, I'll talk about an overview of conservation genetics and the program I run at the Missouri Botanical Garden. Okay, so the theme of the conference was rarity, and so I felt I would be remiss in a talk about rarity without talking about the 7 forms of rarity by Rabinowitz. So this is a paper that tried to define the factors that that shape rarity in plants or animals. And they defined rarity according to 3 characteristics. So it's geographic range size, habitat specificity, and then local population size. And so you see these 8 boxes. Well, so only one of these 8 boxes is actually a common species. So that's a species that have large geographic range sizes, wide habitat specificity, and then large dominant population sizes. And then all the other categories have at least one characteristic that makes the species rare. So they may have a combination of 2 characteristics, which usually means they're more rare. And then the most rare category is species that have a narrow habitat specificity, a small geographic range size, and they have small population sizes. And actually several of the species I'm going to talk about today have those characteristics. But one of the things I also want to point out here is that essentially 7 out of these 8 categories are species that are rare. And so there's actually a lot of species out there that are rare. And I think this is illustrated well in a diagram here. So in this, on the y-axis, you have the number of species, and on the x-axis, you have the number of geographical units where a species is present. And what this shows is that there's a lot of species out there that are extremely rare. And then there's just a few kind of on this end of the tail that are actually widespread. So actually, there's a lot of really rare species out there. And the reason that this is a problem is because biodiversity is declining. So the current plant species extinction rate is around 500 times the background rate. And an estimated 42.8% of plant species are listed as threatened. And the cause of this is essentially a competition with a single species, which is us. We do things like destroy their natural ecosystems, we use them unsustainably, and then there's also the threat of future climate change. So how do we prevent species from going extinct? In my opinion, a good way to do that is to conserve genetic diversity. So genetic diversity is important because it produces the physical and functional differences among individuals and populations. And these differences promote resilience and adaptive potential. So for example, if there's a disease and you have a genetically diverse population, there's a chance that there may be an individual in there that can withstand the disease. Whereas in a genetically poor population, maybe there isn't that individual. But I would say that our knowledge of genetic diversity is very limited because it's not something that you can go out and measure. You have to actually collect leaves or collect tissues and analyze them genetically. And it's also difficult to predict because it's affected by things like the demographic history of populations and also things like life history traits that maybe we don't know a lot about. And so that's where I think conservation comes in, or conservation genetics comes in, is that we really need an accurate understanding of biodiversity in order to conserve it. If you don't know what you have, you can't conserve it. So that's what we do. We use genetic tools to quantify both genetic and species diversity and ensure that genetic diversity is conserved. Okay, so this is a flyer that was made several years ago by my lab manager that just shows kind of a range of some of the species that we work on. And we work on things like, big trees, big ebonies in the Mascarene Islands. We work on agaves. We work on these tiny little, herbs in the Midwest. And the main theme of our work is essentially species that are listed, usually things that are listed as endangered or threatened under the U.S. Endangered Species Act for things that are in North America. Sometimes we also work on things that are maybe listed as critically imperiled S1s through nature serve categories. So maybe they're very rare, but maybe they aren't rare enough or endangered enough to be federally listed. And the way that we work is a lot of times we work with specific partners. So people like the U.S. Fish and Wildlife Service or the Missouri Department of Conservation or Florida Forest Service. And they come to us with specific questions that they want answered about their endangered species that can be answered with genetic tools. And then we work together to find funding, and then we do a project to answer specific questions. So they can be questions like, how is genetic diversity structured across the landscape, and how do we do a good job? Are we doing a good job of preserving it in situ, so in its natural habitat, or ex situ in living collections like in seed banks or botanical garden collections. We also answer questions about, for example, what's the relationship between genetic diversity and reproductive success? And are losses in genetic diversity causing low reproductive success? We answer questions like, is something a valid species and worthy of protection, or is it maybe a subpopulation of a more widespread species, so we don't need to conserve it? And then also kind of questions relating to ex situ conservation, so conservation in seed banks or living collections. So for example, how much genetic diversity is present in an ex situ collection relative to wild populations? Or for example, we can use it to design reintroductions to make sure they have high genetic diversity. So we'll specifically select germplasm to make sure it has a good combination and you can avoid inbreeding. Okay, so what do we do? Okay, so what we do is we'll go out into a population, either in ex situ collections or in situ collections, and we'll sample leaves. So this is what one of our collections looks like. So we have a little leaf here. There's silica gel in this, and it dries out the leaves and it preserves them for DNA analysis. We bring them back to the lab. This is what my lab looks like. We extract DNA out of them. And then we will genotype the same DNA regions from different individuals and compare them. And then we'll analyze patterns of similarities and differences among the individuals in those populations and use that to answer conservation questions. So what we'll do is then we'll usually communicate our results either through reports or through talks to land managers. And so they'll use the data for specific purposes. So one may be to identify priorities for in situ conservation. So places where, for example, they want to, acquire a site as a preserve, like Missouri Department of Conservation. A lot of times we'll use that as a guideline for where to purchase like tracts of land for conservation. Or maybe it'll highlight sites where they need management. So for example, if a population is declining and we see that there's a decline in genetic diversity, then we can say, hey, this one needs more management and maybe you need to burn it. So it's kind of a way to identify priorities. Another thing that we can do is use the information to guide ex situ conservation efforts. So for example, I'll communicate my results to my coworker who has a conservation and seed bank. He's in charge of that. And he'll use it to devise a strategy to make sure that we're getting all the genetic diversity that we can in these ex situ collections. Okay, and then finally, we'll use it to guide selection of germplasm for reintroductions. So, for example, we want to make sure that if we have a very small number of parents, that we take the germplasm from all of those parents to make sure reintroductions have as much genetic diversity as they can. Another thing that we'll do is it's used for conservation policy. So we work a lot with the U.S. Fish and Wildlife Service, which is the agency that's managed with with federal listing and making decisions about whether things are ready to be uplisted or downlisted. And so, for example, there's a thing called this species status assessment, and a lot of our information will be used in those, and it's kind of a state of knowledge about a species. And then a lot of times it will also our information will be used to determine whether recovery criteria, which are the things that need to be met for a species to be downlisted, have been met, especially if they deal with genetic diversity. And then another thing we do is help kind of make recommendations for future work for conservation and management. Okay. So next, I will talk a bit about some examples of how we've used genetic data to devise conservation efforts to protect genetic diversity in several rare midwestern species. So this is 3 projects that were all funded by the U.S. Fish and Wildlife Service, and it focuses on 3 different species. So the first is over here on the left, Leavenworthia exigua var. Liceneata. The second is Physaria filiformis. And the third is Geocarpon minimum. So all of these species are federally threatened. And generally they're somewhat similar in that they have similar habitat preferences. So, talking about our 7 forms of rarity, these are species that have really high habitat specificity, and they occupy rocky outcrops and glades. So that's kind of one of the reasons why these species are rare to begin with. But they also are experiencing a lot of habitat loss. And then, with development comes fire suppression. And so that's the 2 probably main reasons why these species are listed as threatened. And they all have a relatively similar life history strategies, or so we thought when we started. We know that they're all winter annuals. So they germinate in the fall, overwinter as a rosette, and then reproduce in the spring. We didn't know their reproductive biology when we started these projects, but we knew that they all had gravity or water dispersal of their seeds. So the questions that we were tasked to answer is just how is genetic diversity structured across the landscape in these 3 species? What's the best strategy to conserve their genetic diversity? And then are we doing enough to delist these species? Several of them, Geocarpon and Physaria, that was kind of the whole goal, was they thought they were going to be able to delist these species, but they just need to do kind of a little final check. So that was kind of the main goal here. Okay, so this is the first species. This is the Leavenworthia exigua, and this species, one of the reasons why this one's endangered is it has a very narrow geographic range. So that's another thing that makes it rare. It just occurs in 2 counties south of Louisville, Kentucky. So Louisville's right here, and it occurs right here. And then there's another species, which is right here. So this is Leavenworthia exigua var. Liceneata, not another species, another variety, sorry. So this is a variety Liceneata. This one right here is a variety exigua. This one is listed as federally threatened. This one is unlisted. So what we did is we went out and we sampled from each one of these dots here, these dots in the map. And then it's also 5 populations from here. And we we sampled 24 individuals per population and genotyped them. And then we looked to see patterns of genetic structure. Okay, so this is what we found. So this is a genetic distance tree. So each one of these little things represents a population that we sampled. And the branches represent the genetic distance between them. So how genetically similar they are. So the ones on shorter branches are genetically more similar to each other, and the ones that are farther apart are more genetically different. And so I'll point you first to these populations. So this is the variety exigua. This is the variety that's not endangered or listed at all. And what we found is that you see there's branches here. So essentially we see that there's differences among the populations. The populations are genetically different from each other. And then when we looked at variety licineata, which is here, what we found is that the populations are almost all genetically identical to each other. When we look at the genotype data, almost every single individual is genetically identical. And we think the reason that this is happening is because the species we think is apomictic. So apomixis is asexual reproduction by seed. So essentially what it does is an individual makes a genetically identical copy of itself. And then that seed is able to be dispersed across the landscape. So essentially this, this whole variety is essentially like one genetic clone. So they're all the same. But in some ways that makes it really easy to conserve the genetic diversity in this species, right? Because this is essentially you can sample or protect one population and you've protected the entire genetic diversity in the species. So for this one, in terms of genetic diversity, it's easy, but, we want to conserve evolutionary potential, not just necessarily genetic diversity. And so for for variety licineata, we've kind of revised our conservation goals. So we want to protect the evolutionary potential. And I think the best way to do that is maybe to conserve the largest populations, because that can essentially increase the chances that there may be some residual sexual reproduction that can create some variation. And then the other thing that we're doing in this is investigating other types of variation. So things like phenotypic variation. So for example, differences in flowering time, and then also epigenetic variation, which is kind of a type of variation that doesn't involve changes in the gene sequence, but it changes gene expression. So those are the two things that we're looking for. And actually these recommendations were incorporated into the recovery plan for the species. I think it might be one of the first recovery plans to actually mention epigenetic variation. my former graduate student's really proud of that. So I thought I'd mention it. Okay, so the next species I'll talk about is Physaria filiformis. This is this little species here, Brassicacea, and this one has a relatively disjunct geographic distribution. So this one, there's a group of populations that occur in Southwest Missouri up there, another two populations that occur in Northwest Arkansas, then there's two in North Central Arkansas, and then there's a group of populations down in the Ouachita Mountains of Arkansas. So a relatively discontinuous geographic variation. And talking about the forms of rarity, this species has a relatively narrow geographic distribution, and it has oftentimes very small population sizes and very high habitat specificity. Okay, so for this one, once again, we went out and we sampled from each one of those little dots that you see on the map, and we sampled 24 individuals, genotype them, and look at patterns of genetic structure. So for this, what we found was something completely different than what we found for the last species I just talked about. So this is a structure diagram. So this is what I'm talking about here is structure. So each one of these lines represents an individual. And then the colors represent genetic groups. And then these blocks of individuals here represent populations that we sample from. And essentially the way this program works is it assigns individuals into genetic groups based on a metric called Hardy-Weinberg equilibrium. And so essentially what it comes up with are essentially genetically cohesive groups. And so what we found is that there's very strong genetic structure in this species. So for example, these are the populations in the Ouachita Mountains, they all form a genetic group. This one in Northwestern Arkansas forms a separate group. These ones in North Central Arkansas form a group. And then this inset shows Missouri, and we see that there's geographically structured groups in Missouri at a relatively fine geographic scale. So the reason why we think we're seeing this is this species is actually self-incompatible, but it's insect pollinated. And so what's happening is, and it has gravity or water dispersed seeds. So what's happening is that those pollinators are just not able to move pollen very far across the landscape, and the seeds are dropped right underneath them. So we get a lot of genetic structure in this group, in this species. So in terms of conservation, what we want to do in terms of protecting this neutral genetic variation is protect each one of these groups. And so actually for this species, we're doing a pretty good job. So these arrows that you see here are populations that are federally or state preserves. And so almost all of the populations are protected except for these ones right here. Actually, I think these ones are here. So those ones are priority for conservation. So there's about 5 populations there and none of them are protected. They're all in private lands. And another thing that we found in this group, so this is once again a genetic distance tree like I showed you before. So each one of these little labels here represents a population. The branches show the differences among the population. And what we found here is that all of the populations here, these ones in northwestern Arkansas, north central Arkansas, southwest Missouri, which all occur here, here, and here, are relatively genetically similar to each other. But we sampled an outgroup, which is here. This is Physaria gracilis, that's circled in red. And what we found is that the populations here in the Ouachita Mountains are as genetically different from the rest of Physaria filiformis as the outgroup. And so after that, we were like, well, I wonder what's going on. So we've done some additional analyses to look at these populations, and we think that they're, so we looked at, for example, they have they occur in different substrates. So these occur on shale glades, so the shale substrate, where all the rest of the populations occur on calcareous substrates. They're in a different physiographic region. So these populations are all Ozark Plateau, and these are the Ouachita Mountains, and they're separated by the Arkansas River Valley, which is not appropriate habitat. And they also have some morphological differences, very small changes in petal morphology. And it looks like they diverged at least 15,000 years ago, which is actually not that long, but if these are annual species. So that means they've been separated for 15,000 generations. So we're working on describing it as a new species called Physaria ouachitensis. But in terms of conservation, so this is a problem, right? The species is already, Physaria filiformis is already federally listed. And so what that means for Physaria filiformis is that the range of its geographic range is much smaller. And essentially it's like, 5 fewer populations. So in fact, it's probably more endangered than we originally thought after we named this as a distinct species. And then we think this is also probably going to be listed as endangered. There's only like 5 populations and only one of them is really well protected. So that's really kind of changed the conservation landscape. And unfortunately, this is one of the species they wanted to delist, and now we don't think it's a good idea at this stage. Okay. So the last is a species called Geocarpon. And it's funny because that picture makes it look really big, but it's about the size of a dime. It's this itty bitty little plant. You have to crawl around on your hands and knees to find it. And this actually has a relatively wide geographic distribution going from Missouri all the way through Arkansas, Texas, and Louisiana. But it's extremely rare where it occurs. So it's got a high habitat specificity, small population sizes. So this species has 2 habitats that it occurs in. In the northern part of its range, it occurs in sandstone glades. So it occurs in these little depressions in the rock and the sand. And then in the southern part of the range, it occurs in these saline barrens. It's these areas that have high mineral content, and the plants live in these little, they call them like slicks. And it's like areas that are devoid of vegetation, and these Geocarpon will live there. And so we thought perhaps since there's 2 habitat types, we might see some genetic variation structured according to that. So once again, what we did is we sampled populations at each one of the dots, genotyped them, and looked at patterns of structure. And so for this, what we found, so what we found is that in each population, so you see each one of these, it essentially contains a single color. what we found is that almost every individual in each population is genetically identical to each other, and they're very homozygous, which essentially means that they're self-fertilizing. And so the way that this species works is it essentially self-fertilizes, drops genetically identical seed underneath it. But that self-fertilizing causes what's called really strong genetic drift, which causes divergences among populations. So essentially what we found is that, for example, these two populations here, which are right next to each other, they're like right across the road. They're completely genetically divergent from each other. So essentially the way genetic variation is structured in this species is every single population is genetically unique. So in order to conserve this one, you basically have to protect every single population if you want to do a good job of conserving their genetic diversity. And for this one, the recovery plan for delisting states that you need to protect the range of genetic variation. And right now there's only about 16 of the 40 populations that we know of are protected. So essentially about 40% of the genetic variation is conserved. So once again, probably not a good candidate for delisting yet since they haven't met the recovery criteria. And there's a couple of regions, both Louisiana, like 0 populations in Louisiana are protected. And then Missouri, only 7 of the 22 populations that they know of are protected. So those are the priorities for those species, or for that species. Okay, so essentially I showed you these 3 examples because we thought we were going to find very similar patterns of genetic structure in these 3 species, given a lot of the similarities among them. And what we found is that we got completely different answers in terms of how we should protect genetic variation. The first one, you could protect one population. The second one, you could protect, a subset of populations. And the third one, you need to protect every population. And so in some ways, it's important to do a genetic study just because we don't know that until we go out there and find the information out. And I think it provides very practical information for guiding efforts to conserve genetic diversity. And then these information that these data that we've generated are directly being used in terms of their conservation, so integrated into recovery plans and being used to make decisions about listing status. Okay, so next, I'm going to talk about a project where we've used genetic tools to understand the relationship between genetic diversity and reproductive success. And this is a project about Mead's milkweed. So Asclepias meadii. This is a long-lived perennial species, and it's generally found only in mature until tallgrass prairie and glades. And actually, there's only a specific glade that it's found in, the rhyolite glades in southern Missouri, and near the St. Francis Mountains. And generally, in terms of the forms of rarity, this has a relatively large geographic distribution, but has very high habitat specificity, and it has very small population sizes. So it has many of the different forms of rarity. This species is bee pollinated and wind dispersed. And another thing that's really important is that it's clonal. So you'll see a cluster of individuals, and they'll be connected underground via their roots. And so if you take a leaf tissue from multiple individuals, you'll find that they're all genetically identical to each other. So if you go out to a population, you may see like 50 stems, but it may be that there's only 20 individuals that are actually there. It's also self-incompatible. So it has a genetic system that prevents self-fertilization and also reproduction between close relatives, which is a lot of times clonal species will be self-incompatible. And so this species is really rare and endangered, largely because of habitat loss. So this is predominantly found in tallgrass prairies. And as you probably are all well aware, the tallgrass prairies are essentially a very, very endangered ecosystem. So I think about only 4% of estimates say that 4% of the tallgrass prairie remains. And so in light green here, we have the original extent of the tallgrass prairie, and in green is the actual remnant tallgrass prairie. So the species has become endangered just because of pure habitat loss. A lot of the areas have been tilled and then used for agriculture, and so there's just very little habitat remaining. And we think, so the species used to occur, I think, relatively widespread across the Midwestern US. but it's declining. So it's, I think, completely gone now from Wisconsin and Indiana. There's just a few small populations that remain in Iowa and Illinois. And actually somebody was telling me about them earlier. And then the main part of the range is in Kansas and Missouri, and it's also declining there. So it's suffering from a lot of habitat loss and declines. And so because of that, this is also listed as federally threatened. But there's another reason why it is threatened, and that's because it has very low fecundity. So essentially, one of the main ways to manage a tallgrass prairie is haying, and this is something that happens usually in midsummer. But Mead's milkweed flowers in May, and then it takes like 3 to 4 months to produce seed pods. So you can usually collect the seed pods in like September. And so if you hay in midsummer, essentially they just cut the seed pods off and it doesn't reproduce. But the other thing is that even in populations that aren't managed by haying, there's a lot of populations that will produce a bunch of flowering stems, but it will never actually produce seeds. And that's a problem, obviously, because you need reproduction for a species to be self-sustaining. And there's also a lot of variation in reproductive success. So there's usually a couple populations out there that will actually produce seed pods. And then many populations will produce like little to no seed pods at all. And so that's actually where I came in, is that a colleague wanted to know why, basically. So there's always been a hypothesis. There's a hypothesis that was asserted in the literature a long time ago about the cause of low fecundity. And it's essentially a genetic hypothesis. So essentially the idea is that the fragmentation of the prairie, declines in population size, and then loss of genotypes in the population have essentially gotten to the point where you just have very few genotypes. And because the species is clonal and self-incompatible, this has led to the point at which the species essentially has no available mates in the population. So essentially it can't reproduce because there's no compatible mates. Because of this theory that was advanced in literature, there's actually been quite a bit of management that's been done to address it. So for example, the Missouri Department of Conservation has done this augmentation program where they've transplanted over 1,000 seedlings into 8 prairies all across the range in Missouri, kind of under the assumption that genetic diversity was the cause of the low reproductive success. However, it's a little unclear. There was never actually a study done that would test the relationship between those two factors. And so, the question is this actually going to have its desired effects? And then the other thing is that should we be moving individuals across regions? There's a thing called outbreeding depression, where you may have a locally adapted genotype. You mix it with a different locally adapted genotype, and then you get these offspring that are not adapted to anything. So there's a big problem if genetic diversity is not the cause. So essentially that's what I was tasked to address. So what we wanted to do was look to see if there was a relationship between low genetic diversity and low fecundity in the species. So essentially what we were looking at is a relationship between fitness and then a bunch of genetic parameters as well as demographic parameters. And then also we wanted to know how we could manage populations to increase their reproductive success. Okay, so what we did, so this is a map showing our study site. So over here is Kansas City, and over here is St. Louis. And then each one of these little sites on the map in red are our study sites. So we essentially drove all over Kansas and Missouri for this project. And essentially what we did is we mapped, we tried to find every single stem in a population, and we mapped it and we tagged it. We sampled leaf tissue for a genetic analysis. Then we genotyped all the samples, and then we also did a kind of thorough demographic study. So we essentially revisited the population, recorded individuals if they're flowering or fruiting, bagged the seeds, and then also collected and counted seeds for an estimate of fitness. And then we essentially just looked at the correlation between fecundity and genetic diversity and other demographic parameters. All right, so this is the results of our fecundity analysis. So on the left here are 12 population sites. And And this is the number of stems that we sampled. Those are the number of stems that were present in each population. And as you can see, so down here, it shows the total number of individuals that flowered. So about 24% of the individual stems that we surveyed flowered. But if you look at the fecundity, I'm blocking right here. So essentially what we found is that only 43 out of 1,160 stems set seed. And we saw a very wide variation in reproductive success. So almost all the populations that you see here produced only 0 or 1 or 2 fruits. And then there was 2 populations that produced the vast majority of seeds. So 27 seed pods here and 11 here. So as people have mentioned before, we saw a lot of variation in fecundity with a few populations producing a large number of fruits and then most of the populations producing 0 fruits. Okay, so when we looked at genetic diversity, though, we found patterns that were actually not what we were expecting. So once again, this is the number of stems that we sampled here. And this is the number of genetically different individuals in the population. And one of the things that we were surprised to see is that even in the small population, so for example, there's this one here that had 8 individuals, we had 4 genetically different individuals, which is actually enough to reproduce. And also we saw relatively high genetic diversity. So these are estimates of genetic diversity. Those are high. And we also saw really no inbreeding. So this is an inbreeding coefficient, and essentially 0 means there's no inbreeding. And essentially we saw little to no inbreeding in these populations. So this is actually kind of opposite what we were expecting. We were expecting to see that the smaller populations would have much lower genetic diversity and maybe higher inbreeding. And when we looked at the genetic structure, so once again, this is a structure diagram. And so each one of these lines represents an individual and the colors represent genetic groups. And what we found is that there's virtually no genetic structure. So we were expecting each population to look different from the next one. And what we found is that individuals in one population may be more closely related to individuals in a different population than they are to other individuals in the same population. Which was completely counter to expectations. And we find that regardless of how we divide up or the number of groups we divide up individuals into. So this was also not expected. And so, based on these metrics, we looked at the relationship between, let's see, so the number of fruits is our estimate of fecundity here on the y-axis. And then these are different metrics of genetic diversity. And essentially what we found is there was no relationship between genetic diversity and reproductive success. So this long-standing theory was not supported by our data. So what we found is, populations are very genetically diverse and they contain multiple unrelated genotypes. And we think that the reason why this is happening is that I think these plants can probably live 50 to 100 years. And so what we think has happened is that we're seeing the patterns of genetic structure from 50 years ago when there's big swaths of continuous prairie. Also, the species is wind dispersed, so it has seeds that can blow into the wind and go several hundred miles. And so essentially we're seeing the genetic structure, characteristic of a wind dispersed species. But the kind of takeaway message is that the low genetic diversity is not the reason why the species is exhibiting low reproductive success. So essentially our data completely disproved the theory that's been advanced in probably 15 or 20 papers that I've read in the literature. So what is the answer? So what we found is that the only demographic characteristic that was significantly associated with fecundity was actually much more simple. Essentially what we found is that populations that had a large number of flowering individuals had a high reproductive success. And we actually found that there's kind of a threshold effect where if you have more than 50 flowering individuals in a population, you get a much higher rate of reproductive success. So essentially, the more flowering individuals, the more fruits. Much more simple. I think people like to think that it was a genetic study because they didn't, it's kind of a black box, but it turns out that it's much more simple. And so this pattern that we see here, where you see a threshold effect, is actually kind of a characteristic of something called an Allee effect. And this is an effect where the aggregation of individuals is essential for population growth. So you can see here, this is kind of what it looks like. So this is the population size along the x-axis, and this is the population growth rate. And in a species with a strong Allee effect, what you'll see is that in small populations, you'll actually get a negative population growth rate until you reach a specific size, in which case you get an increase in population growth rate. And this is in contrast to a species with a no Allee effect. And there's several things that can cause Allee effects in plants in small populations. So for example, small populations may have a lot of inbreeding depression or loss of genetically compatible mates, which was the hypothesis that we were testing. We disproved that one. There's another kind of category, which would be ecological Allee effects. So sometimes larger populations may have less predation pressure, just because there's more individuals, less predation per individual. But we didn't see any evidence of that. And then the last one is a reproductive Allee effect, where essentially you get either reduced pollinator attraction or reduced ability for pollinators to transfer pollen among closely related or between individuals of the same species. And this is what we actually think is happening here. So we think that the Allee effect is essentially related to pollination, where essentially you just have to have a certain number of individuals for pollination to occur. And we think there's two underlying causes. One could be pollinator limitation. Essentially, there's just pollinators are just not attracted to small populations. There just might not be enough individuals aggregated to attract pollinators. Or it could be a pollen limitation, where pollen just isn't transferred among conspecifics. So I don't know if you guys know about the pollination biology of milkweed, but in some ways I think that it kind of supports the second hypothesis here. So in milkweeds, the pollen is packaged into a pollinarium, where you get these two pollinate sacs, and they're connected by this thing called a corpusculum right here. And you can see there the corpusculum sits right there. And essentially what happens is when a bee visits the flower, the corpusculum will get stuck on the bee's leg, and then the bee has to transfer it to another individual and then get the pollinarium into this thing called a stigmatic slit right here. So honestly, I find it amazing that any pollination occurs in any milkweeds at all, just because it's such a complex pollination system. So in some ways, I think that perhaps there's just pollen limitation. Pollinators just can't get the pollen where it needs to go in a small population. But this is something that we're hoping to test in an additional study that hopefully we'll hear about funding on in the next few weeks. Okay, but in terms of management of the species, in order to get reproduction occurring, basically the goal is to reach 50 flowering individuals in a population. So there's a couple things that we recommend. So the one is that, so there's certain populations out there that basically just will never have 50 flowering stems. They don't even have 50 stems at all, right? There's a lot of populations in Illinois. I was talking to somebody today that, there's 10 or 12 individuals. So for those populations, we really need to just increase population size. So we can do augmentations of the population. And it's interesting because the whole reason why we did this study is because we wanted to know if this augmentation program by the Missouri Department of Conservation was the right step. And it turns out, they didn't have the right underlying reasons, but they were actually doing the right thing. So, but they didn't know that really the goal was to get 50 flowering individuals. So now we have a better target, a more clear target. And actually, so we've actually started doing this. So a lot of the seeds that we collected as part of our study, we germinated them, and we've done several, we've done a augmentation. So this is a barrier conservation area. And then here's an individual that was actually flowering from the reintroduction that we did right there. The other thing is, to burn. So in milkweeds, when you burn a population, essentially what happens is you get a burst of flowering. So you burn in the dormant season, you get a burst of flowering the following summer. And so fire really synchronizes flowering in this species. And so what we've recommended is to start burning larger tracts of prairie at once. So if you think about kind of the way that the prairie was spaced across the Midwest, there are these large continuous swaths of tallgrass prairie, and Native Americans would burn them. And we think that there's large tracts that would get burnt at once. And so now what happens is each little remnant prairie is managed in its own way, and they have their own burning schedule. And so there's this really hodgepodge kind of mishmash like management going on. And so what we recommended, and we actually talked to land managers about this, is to burn all the populations at once so we can get a synchronization of flowering. Or burn as much of a population as you can, coordinate between neighboring sites to burn them all at the same time so that we can get a synchronization of flowering, so we can get that critical threshold of 50 individuals flowering at once. Okay, so overall, in conclusion, conservation genetics, I think, in all these cases, they've provided unexpected and I think important insights about biodiversity, and it provides very clear information about species management. And overall, I just want to talk about how you can help conserve rare plants. Just a few ideas. So for example, voting, right? Trying to get people into office that are conservation minded is a really important one, probably one of the most important things that we can do. Second is volunteering to help with habitat restoration efforts. So for example, honeysuckle roundups, trying to improve habitat is really important for all of those rare species out there. Let's see, native plants, using native plants in landscaping to promote, the pollinator health. And then finally, collecting data about rare plants. I think a lot of you guys out there are really knowledgeable about your plants. So if you see a population of a rare plant, collect some data, collect, population size estimates, photos, take population counts, directions, and then either document it on iNaturalist or submit it to the Natural Heritage Database. A lot of populations in natural heritage databases haven't been updated in 20, 25 years. And if you're out there and you see a population of a rare plant, help us learn more about it, because we rely on a lot of those records. Okay, so with that, I just want to thank all these people, and thank you.